Mohammed Umar Farooq biological video. Please make sure to subscribe, like and share. Ask the question for answer and comment about the artwork. You can also answer question to each other for solving the problem in the comment below. So today we will discuss about the virus structure, function and shape, and there are some important character. So the virus is the basically non-cellular infectious particle. So there are many viruses in shape and structure on the basis of genome and shape. So the non-cellular are infectious particle. So the virus is the non-living in the outside of the host. It means it is a parasite. It's basically depend to the host, and it is non-cellular part, non-living, and their character is living inside of the host, while it is infectious particle contain DNA or RNA as a genetic material. So it is genetic material used for the replication inside of the host and reproduce only living organism. So their reproduction is depend on the host into the host. So the disease causing agent it is smaller smaller 28 nanometer, while the larger virus can be 250 nanometer. So it is visible electron microscope and able to pass filter. So it can pass filter. So here is the general structure of the virus we can understand well. So here is the DNA inside is covered by the capsid. Capsid is the protein is made up of smaller protein peptides, while the genome are present inside. Genome is known as the DNA, RNA, double-stranded, single-stranded. We will discuss. So the whole infectious particle with genome and protein is known as viron, while the upper part is known as the spike protein. This spike protein is important character for reception of the host cell to invade inside of the host cell for replication. So important to know that if we will study under the electron microscope, we can check the DNA structure and DNA shape and their types of DNA. So the DNA virus basically genome is made up of different types: a DNA virus, double-stranded DNA virus, RNA virus, or double-stranded RNA virus, or single-stranded RNA virus, or some other is a supercoiled and double-stranded DNA and relaxed double-stranded DNA, as well as DNA, B double-stranded DNA. So here is the DNA virus which contain DNA, while here is the RNA virus which here is the double-stranded, and here is the double-stranded DNA. It means the two DNAs interconnect to each other, while here is the RNA virus which that is contain only RNA, and double-stranded RNA mean the two strand of RNA are present in here, and here is the double-stranded DNA is present, and that is supercoiled and relaxed double-stranded DNA. So the here is the relaxed double-stranded DNA, and here is the B double-stranded DNA are present. So these genome are present in the different type of virus. So let's talk about here is we can understand that is the capsid is made up of capsomere. So the capsid is the outer protective coat of the bacteria which that protect the genome. So it means the capsid is made up of capsomere, and on the other hand, we will discuss about the type of virus. So the different type of virus we can not cover the shape of the virus in this video, but we can just overview about that. So the first of all we will discuss on the basis of host. So it is animal virus. So mostly animal viruses contain DNA, and animal virus host specific. It means the virus is specific for the host to infection and infect. So it means the spike protein contain which that is receptor, and this spike protein is unique for a unique host, and it is made up of capsid, and inside is the DNA. While here is the plant virus, we can understand the plant virus is basically mostly plant viruses contain RNA. So these virus infect to the plant, and it is infect to the different plant on the basis of different specificity on the spike protein are present in the protective protein coat on the upper side. So example of the plant virus is known as a tobacco mosaic virus, which that cause the disease on the tobacco plant. Uh, it is also known as DMV. While the retrovirus is basically, we will discuss in this diagram, the retrovirus is depend to the human virus. It is not depend to, it is not infect to the apes, because they don't, they have not contain CD4 molecule on the T lymphocyte. So it means the T helper cell contain a CD4 receptor. So that CD4 receptor spike protein are present in the upper side, and here is the inside is the double two RNA, double stranded two RNA are present, and reverse transcriptase enzyme and diploid RNA are present. Diploid RNA is present. So the AIDS causing virus, and some virus is the hepatitis virus, hepatitis C, hepatitis B, hepatitis A is the character of this type of retrovirus. So the RNA, it is the set of RNA, reverse transcriptase, integrase, and polymerase are present inside of the virus, within the capsid. So it means the virus is here, which that is recognize the CD4 molecules and CD4 receptor, which that receive that and insert inside. So and insert to the T lymph helper cell to kill the leukocytes. It means the loss of immunity, and that cause the different acquired immunodeficiency. So virus is another type, which that is a plant virus and contains single-stranded DNA virus. It means that single-stranded DNA virus is looking like this. So let's talk about the shape of virus. Shape of virus on the basis of their infectious particle. Type of virus for infection. So the infection side we will cover in this diagram to understand the all virus. So here is the papilloma virus, which that cause the disease by the sexual contact or blood transfusion, contaminated blood transfusion, shaving, or some other reason. So the sexual contact is the major example. So here's the pox virus are present, and that pox virus structure is look like this. While the herpes virus, which cause herpes disease, herpes simplex disease, which that is the rounded, and that is additional lipoprotein are present in the upper side on the capsid. So it means here is the here is the another virus is the basically hexone and pentone are present, and the pentone base, and this virus is the cause common cold, flu, and mm, uh, acute bronchitis and pneumonia, while the single-stranded RNA are present in the COVID-19 and SARS and MERS, severe acute respiratory syndrome, and Middle East respiratory syndrome. It is contain ACE2 spike protein, which that is the used for the receptor of the epithelium of lung cell. So it cause the disease on the lungs to shortness of breath. So here is the alpha mosaic virus, which that cause the disease on grass, while the mumps is the another virus which that cause disease, and mumps virus is looking like this. So that's mumps virus is cause disease. So here is the adenovirus. Adenovirus structure is like this. It is the polyhedral. So the polyhedral mean the many capsomere are present on the head. So looking like this. While here is the T-even phase. This T-even phase is the bacteriophage, which has complex structure and infect to the bacteria. We will discuss later in next video. So we should understand just here, this is the bacteriophage. It is contain a T3, T4, T5 and T1, which that is the type of T-even coliphage. So here is the potato virus, which that infect the potato plant to cause the disease. So it is can be filamentous or complex. It look like this, and head is contain a genome in the bacteriophage. So here is the influenza virus. Influenza virus contain a spiral DNA where additional lipoprotein and enveloped are present. So it means the additional lipoprotein are present also. So here is the we can say that is the structural part. So here is I am drawing that is a vertex, and that's capsomere is the unit of capsid. So that capsid is made up of capsomere, and that is the polyhedral structure is looking like this. While here is the here is the bacteriophage contain here is the other bacteria which contain a capsomere is made up of capsid, while inside is the spiral coil of RNA. So inside is the spiral coil of RNA are present, so blue color. And bacteriophage contain a head region to contain DNA, which that is infect to the bacteria. So let's talk about the identification of positive viral infection in the living organism. So let's talk about here first of all pre-analytical process in which we can we can collect the sample from the infective person, from the mouth, from the throat, by the by the sample collecting instrument. So the storage and carrier. So after the storage and carrier contain a virus. So this virus is basically we will further process analytical complementary DNA and reverse transcriptase polymerase chain reaction will be used for it, and to produce RNA extraction first of all. So the RNA extraction to degradation by RNases. So the RNases is the enzyme which does degrade into the smaller segment of this RNA, and thus reverse transcription will begin, and reverse transcription to produce a DNA. How it's possible here we will discuss. So the primer is used as a probe. So the probe is the inflorescence containing sample which they are used for the checking after the replication of the viral DNA, to show the inflorescence, to check the either more or less DNA segment, to check the infection of that positive or negative. So here is the RNA, which that is a segment of RNA which bind with the probe. Probe contain primer. So this primer will be bind, and annealing process as extension to 72 degrees Celsius, and annealing is the 52 degrees Celsius to reverse transcription, to begin the reverse transcription of RNA into the DNA, and viral RNA sequence amplification in thermal cycle. So the thermal cycle is used for the polymerase chain reaction to produce copy of that genome. So this genome contain either probe, which that is a specific primer which is mixed with a master mix and nucleotide and other nutrients to basically extension process to make a copy of this DNA segment. So this DNA will be formed, and after the cycle will be continuous thermocycler used for the polymerase chain reaction to produce. So this is the analytical post analytical process in which the check the fluorescence. So here is the PCR cycler, and here is the fluorescence. So here is the baseline, and here is the threshold. When pass out the threshold and move the more fluorescence by the producing of more cycle, so it means the more DNA will be produced. If the fluorescence is more, so it means the positive, while the negative. So thanks for watching. Please subscribe the channel.